Okay, I guess we'll get started. So this will be a general introduction to resting state fMRI. So first we're going to discuss some background and motivation, then some of the more common techniques for analyzing the data. We'll cover some key points relating to interpretation and then touch on a few future directions. So what's resting state fMRI? Well, task-based fMRI is when we're trying to probe some specific aspect of brain function by presenting stimuli or behavioral conditions to the subject at known points in time. And then we focus on the brain activity that's being modulated by that task. So like in this kind of cartoon example, we might be interested in the fMRI signals that have higher levels when people are opening their eyes compared to when they're closed, such as in the visual cortex. Now, in a resting state scan, we don't present any task or stimuli at all, okay? So we're just going to start the scan and record whatever activity happens to be going on at the time. So this might be some example time course that you'll measure at a voxel region or interest in the brain, which you can see doesn't have any particular temporal structure defined by an experiment. So in these scans, typically we instruct the subject to just either keep their eyes closed the entire time or keep them open and fixate on a crosshair. Pretty minimal instructions, maybe not think about anything in particular. And the duration of these scans is usually on the order of 5 to 15 minutes, sort of similar to the length of most task scans, though people sometimes scan in resting state for much longer depending on what you're looking for. And we'll discuss that more later. So why would we want to do this? So why would we want to scan someone when they're not doing anything? Well, one motivation is that, of course, the brain is always active, even when you don't have some experimenter giving you something extra to do. And it's, in fact, been shown that the so-called spontaneous activity actually accounts for the majority of the brain's energy consumption. And if you do a task, it only adds a very small fraction above this already large baseline. And I apologize, I forgot the reference. I'll put it on the online slides. And so therefore, it's possible that we might learn something very useful about brain function by studying this kind of ongoing activity in itself and how that activity is fluctuating over space and time. But then how would we actually go about trying to extract information from this kind of data? You can imagine there might be some challenges because, in task data, we know when our conditions are happening and when they're being presented. And we have, well-defined reference points throughout the scan that we can use to interrogate our signals, but we don't have that here in resting state data. And since we don't have such a reference, it's also harder to know which part of these fluctuations is relating to neurally driven activity rather than artifacts. And importantly, even if we could remove all of the artifacts, it's difficult to nail down exactly what that activity is corresponding to, since we are not measuring any particular behaviors or probing someone's thoughts, and we don't have a clear idea of what kind of other processes might be going on without collecting other measurements. So we'll revisit these central issues later on, but first I wanted to go over some points relating to analysis. So since we don't have a kind of task waveform to use to analyze our data, one of the major ideas that's emerged is to instead look at the relationships between the time courses of different parts of the brain, and this is an idea known as functional connectivity. So in functional connectivity, the basic idea is that we're looking for similarities between the time courses of different brain areas, such as their correlation. So in this cartoon, you could say that these three areas are having very similar time courses, so we'd say that they have high functional connectivity, and so do these two, but the connectivity between them looks a bit lower. From a neuroscience perspective, one motivation to do this kind of analysis is that if areas are indeed showing similar activity, then this could give us some clue into which areas might be having some interaction between them or might be communicating as networks or circuits. But something that we have to keep in mind here is that this functional connectivity analysis is only looking at statistical similarities in these time series. We're not directly looking at the physical structural connectivity, and we're not actually even directly testing that these areas are interacting. So we just have to bear in mind that the interpretation is not that straightforward. So what do you think would happen if we looked at functional connectivity in resting state data? Would we imagine that there are certain areas that show correlations with one another? Well, the answer is yes, and this was the first study that really showed this and launched the field of resting state fMRI. So this was by Bharat Biswal in 1995. And what he did was to have subjects do a task. So he scanned people while they were doing a two-handed finger tapping task. So you get activation in the left and right motor cortex. And then he scanned people in resting state, and he made sure that people weren't moving their fingers by recording EMG. And what he saw was that if you take some voxels in the left motor cortex and you extract their time course, maybe it looks like this, then it turns out that the most correlated regions in the brain happen to fall in other areas of that motor network. So here would be, for example, the time course from the right motor cortex. And so this was really intriguing because it looks like even if you're in resting state and not doing any particular task, and if there's no overt motor activity, this motor network is still most highly correlated with itself compared to with the rest of the brain. And so potentially we may be able to identify functional networks just by looking at their correlations in resting state. And this was shown to generalize to a number of other brain networks. So if you look at the right and left auditory cortex, for example, these were highly correlated in resting state, and so were voxels within the visual cortex, and so were frontal and parietal regions that tend to be involved in functions like working memory. And this study did a direct comparison to task data and found that these patterns do align very well with systems that have been characterized with tasks. So if we think about this, it's pretty incredible because, the areas in these maps came out grouped just by looking at their temporal similarity in rest, which suggests that we may be able to map out multiple functional networks using resting state data alone. And not only that would that make things easier, but it would also allow for studying the integrity of these networks in populations where tasks may not be feasible, such as perhaps very small children or in different states of consciousness. So a couple of notes on the terminology. These patterns are referred to as resting state networks, or sometimes also intrinsic networks, and they refer to sets of regions or nodes that have mutually high connectivity in the resting state. And they're often given names based on the areas that they overlap with. So this one might be called salience network or visual network. But we have to be careful because the terminology is definitely not standardized across the field. And so, you might see a similar looking pattern in two different papers, but it's referred to by a slightly different name. And also the term network tends to be loosely used. So what exactly a paper will mean by a network you'll have to see what they actually did in the analysis to derive that. Most of the literature tends to center on this kind of spatial granularity where there can be identified about 10 to 15 patterns that tend to be very reliably recovered across individuals and over scan sessions. But we can also choose to study functional connectivity across multiple spatial and temporal scales, just as we can with tasks. And we can also further subdivide any of these networks into more tightly connected subnetworks. And so it may not make sense to talk about things like whether there's a fixed number of networks in the brain, since that may really depend on the spatial scale that we're studying. So how reliably can we identify these networks from one scan to another or across individuals? Well, generally, this reliability is very high, and you can see that from these example studies. And so in this one, they acquired multiple resting state scans over several different weeks in the same person, and you can see that these three networks reproduce very well across these weeks. And here's another study where they extracted this pattern called the default mode network from 6 different subjects, each with 2 different scan sessions each. And you can see the same basic topography here across the scans in the individuals. These networks are also quite reliable across different brain states and across different species. And so here's again that default mode network in sleep, as well as in monkeys and rats. And furthermore, these animals also happen to be under anesthesia. So the fact that we can see it in brain states like sleep and anesthesia suggests that the kinds of conscious thoughts and sensations that are going on may not be the main drivers of these networks, and there may be other factors that we'll discuss more later on. So the general picture of these networks is kind of stable, as you can see, across people, but there's also notable variability in their properties across different conditions. And so, for example, if you look at sleep compared to wakefulness, it's been found that the default mode network shows a relative decoupling in its anterior and posterior parts. And we can detect certain differences across neurological and psychiatric disorders. So this early study showed that the hippocampus appeared to have reduced functional connectivity to the default mode network in patients with Alzheimer's disease compared to age-matched controls. And here they found graded changes in the functional connectivity between these default mode network regions as a function of the disease severity in schizophrenia. And as Emily discussed on Friday, there can also be reliable differences between individual subjects. And so this suggests that resting state data may be sensitive to these kinds of pathological or natural changes in brain activity and may therefore potentially yield useful biomarkers. And this has led to a ton of research in this area, many different clinical applications trying to look at resting state features across disorders such as depression or epilepsy, trauma, or migraine. And this table is now very old. It's from 2012, so these numbers are probably like much different now. And I've been trying to find an updated table, and if any of you guys know one, let me know. So that would be great. I wanted to spend a second just talking about the default mode network a little more because it's important in the historical development of resting state. And some of the main nodes of this network are the PCC, or posterior cingulate cortex, the angular gyrus, and the medial prefrontal cortex. And it was pointed out in some early PET studies that these areas had the interesting property of actually showing higher activity during a more passive resting or baseline conditions compared to more externally focused or demanding tasks. And that was different from many other regions of the brain that tend to show more activity during external task demands. And so this helped to encourage more dialogue and research into brain activity that's going on during these kinds of task-free states. And Mike Grecius showed shortly after that the nodes of this the default mode network appeared to be highly correlated with each other in resting state, and so these areas may form a coherent network. And currently, this is one of the most intensively studied networks in the literature. And in addition to having this property of being more active when you're not attending to the external environment, possible functions it's been shown to perhaps have are relating to autobiographical memory or thinking about the future or self versus other types of processing. So to go over some of the points we just talked about, in resting state fMRI, we're scanning people in this passive condition in order to try to study spontaneous brain activity. And even though this condition is very unconstrained, we can still reliably identify a number of spatial patterns of correlated activity that seem to closely correspond to functional networks. And therefore, we may be able to study functional networks without the need for task compliance, and that would open up the possibility to look at a broader range of people and brain states. And since this approach may be sensitive to differences relating to either natural or disease-related processes, then it's now being studied for potential neuroimaging biomarkers. Now I wanted to just cover a few of the methods that are often used to analyze resting state data. These are just three main approaches. There are many others out there, but these are some of the kind of methods that you tend to come across most often. So we'll start with seed-based correlation, and this is very straightforward, and it was the earliest approach to be applied. And it asks the question, which areas are most highly correlated with a specific region of interest called the seed? So the way it works is that you choose some region of interest or voxel called the seed, and then you extract its time series across the resting state scan, and then you correlate that voxel's time series to the time course of every other voxel in the brain, and you can represent every other voxel's correlation with that seed in a map like this, where this is the correlation coefficient. And then you can apply some statistics to threshold that map and come up with areas that are significantly correlated to the seed. So this looks a lot like a sort of standard task activation map, but in this case, the regressor is coming from the seed region rather than your task. And we can still use the GLM framework as usual to implement this. It's just correlations. So some of the example questions that you might ask using seed-based correlation are there any areas whose correlation with your seed is significantly different in one condition versus another, or in patients versus controls, for example? Or maybe you might be interested in whether there are areas whose correlation with the seed region is proportional to some more continuous behavioral or outcome measure, such as medication dose or IQ or something like that. So these are a couple of examples of seed-based maps using several different seeds. This was a seed in the PCC, where you can see that it produces a map that looks a lot like the default mode network. And if you use seeds in the motor or the visual cortex, you also get motor or visual areas as expected. And these are at the sort of typical spatial resolution we've been talking about, but we can also, as I mentioned, do this kind of functional connectivity mapping on more fine spatial scales. And this study looked at the visual system using high-resolution imaging. So when they placed a seed in different areas of visual cortex that corresponded to different eccentricity representations, it looked like the highest correlations in each case tracked to the areas with a similar eccentricity, and even in the contralateral hemisphere. And these color-coded areas were the actual boundaries they had mapped with a task. And this was in the resting state data where the eyes were closed, so it's pretty cool that this organization emerges just from the spontaneous activity. The seed-based analysis is very straightforward, but it does require us to choose a seed. And so when we, where are we going to place it and what size should it be and so forth. And so we're faced with the same concerns that we are when we try to do any kind of ROI-based analysis. So this should depend exactly on what you're studying or how specific your hypothesis is. So some studies will use an atlas-defined region from either an anatomic atlas or perhaps a functional atlas that came from a network analysis itself. Or you might choose some published coordinates in the literature if you want to test a very specific region. And if you want to consider the individual anatomy in more detail, you might draw an ROI manually on a subject's T1, or perhaps do a functional localizer and define your region based on a task activation map. The choice of the seed is really important because this can really impact the results of what our analysis will give us. So for example, looking at the default mode network, this has been reported to subdivide into dorsal and ventral subnetworks, each of which have distinct connectivity patterns. So if you're trying to seed the default mode network and you happen to place your seed more in one subnetwork than another, then you might end up deriving a different pattern of connectivity. And we saw here that this could account for some of the variability in findings across the literature in terms of positive and negative correlations. As another illustration of this sensitivity to the seed location, this is showing what happens when you move the seed along this line between two regions in parietal cortex. So as they push the seed kind of from point A to point B, these were the series of correlation maps that resulted, and you can see that this can kind of change abruptly at some point, even with small shifts in your seed region, if you end up moving between distinct functional areas. And so on the one hand, this is not good because errors in your spatial registration might cause you to end up placing the seed where you don't expect to. But on the other hand, this is actually really interesting because the transition between these different maps could help us identify functional boundaries in someone's brain, which is what they did in this study. So if we're looking for points where there's more rapid change in the appearance of these maps, then that could signify that we're jumping into a different functional area and may let us map out some of these boundaries. And this is a cool illustration of what happens to the seed-based correlation map as you move a seed around different areas of the cortex. So this dot is the seed region, and this was the correlations, thresholded correlations with the rest of the brain. And so you can see how these kinds of continuous changes in the correlation maps that happen. And so this kind of mapping can help us perhaps learn new things about long-range correlation patterns. And as I mentioned, this analysis can really be implemented in any kind of software that'll do linear regression. But here, our AFNI friends have made a very fun and interactive program called Instacore, where you can click on a voxel and it'll show you the correlation map across the whole brain. So this could be very useful for exploratory analysis. So the second method we'll cover is ICA, independent component analysis, which is also a very, very popular method. And the basic idea behind this model is that you input your fMRI data, which is a sequence of bold volumes at each TR across the scan. And what comes out is a set of components that linearly combine at each point to generate the observed data. So in other words, at any given time frame in your data, we can model that as a mixture of activity coming from these different components. So at time 1, you might have A1 times component 1 plus A2 times component 2, and so forth. So these weights can change as you move over time, but the component maps themselves are fixed here. And so these are kind of like the basic units that we say would generate our data. We can also think about each of these components as having a time course that goes with it, which would be defined by the weights of that component at each time point in the data. And so this can be kind of thought about as this component's activity, how strongly it's contributing to the data at each point in time. So by unmixing or decomposing our data into these components, this should tell us a lot about the hidden structure that's in our data. And we tend to get some patterns that look a lot like neural types of functional networks, and others that look more like artifacts, like things around the edges here. And so doing this kind of analysis can help us identify and remove artifacts as well. And just in case it's helpful, the analogy that's often used with ICA is this cocktail party problem, where you can imagine that you're at a crowded party and a bunch of people are talking at the same time, and what you want to do is kind of separate that mixture of voices into what individual people are saying. So here, our data are like the mixture of components that are talking at the same time. So when we run this algorithm, we start with our data, and we don't have any knowledge of what the components are or what their time series are. And so we have to place some assumptions in order to be able to solve this. And one is, the key one for ICA is that we assume that these maps have spatial independence. So we're using this kind of mathematical criterion to try to get these networks, but interestingly, it tends to produce components that look very much like biologically plausible networks, and a lot like the ones that we're getting from seed-based correlation maps. So these are some thresholded ICA maps that they obtained from a group of subjects. And you can identify, default mode and visual network, et cetera. And even though this is doing something mathematically very different from seed-based correlation, it tends to converge on these patterns of areas that have highly coherent activity. This seems to be a very robust feature in our data. So looking at some components and time courses, as I mentioned, you'll get a number that look like networks of interest, whereas other ones may look like artifacts or maybe components of no interest. And this is a nice way of visualizing this kind of hidden structure. And the fact that you get artifact components split off into separate, split off separately is kind of an automatic denoising feature, because it leaves some of these network-like components less contaminated by definition. So some things that people do include choosing components that look noisy and just eliminating them, and then you can either carry these components forward in your analysis, or you can reconstruct the data from the set of clean components and then run other kinds of analyses on it. But in order to do this, we have to have some objective way of identifying those noise components. And so there's been some research in that area. And one approach that's been developed by the Oxford group uses machine learning to try to learn the spatial and temporal signatures of artifact versus neuronal components based on training data. And they have provided some software that will allow you to try to automatically classify them, and that's in FSL. Another technique that's rapidly becoming more popular is multi-echo ICA, which was developed here at NIH in Peter Bandettini's group. And I think next week there will be a lecture all about that. But briefly, the technique requires that you do a multi-echo acquisition, which means that instead of acquiring an image at just one echo time per TR, you're actually acquiring images at multiple echo times. And it uses the fact that bold signals show a TE-dependent scaling in their percent signal change, which is something you would expect in kind of real brain networks, whereas non-bold components would not have this echo time dependence. And therefore, you can do something like rank your ICA components based on how bold-like or non-bold-like they are, and where most of the artifacts would fall in the non-bold end of the spectrum. And so this can be a way of trying to separate those components in a more automatic fashion. But not all artifacts separate out very well with ICA. That's important. So it's not a magical solution. So some, in particular, some physiological effects like relating to breathing and heart rate, these actually arise from bold mechanisms. And so we would need other kinds of methods to try to separate them. So to summarize a few things about ICA, it's multivariate, and you are extracting many networks at once. This is in contrast to seed-based analysis, which is more of a pairwise analysis. And it doesn't require you to choose a seed, but you do need to specify various parameters in this algorithm. For example, the number of components you'd like it to return. This can actually really impact the results that you get, because if you imagine taking a data set and asking for 200 components out, then you're going to get networks and artifacts that tend to kind of split up a lot more spatially than if you're asking for just 30, where things will be lumped more together. And there's some heuristic kind of methods you can use to try to automatically determine good numbers of components, but it's kind of still really tricky. And ICA can be used for both network analysis and noise reduction, as we just discussed. And it's still kind of also tricky to compare results of ICA between different subjects and groups, and this is not completely straightforward, but there have been several techniques that have been developed by the fMRI community to try to handle some sorts of multi-subject analyses, one of which is called dual regression. And just because what we're doing to the data is a little more tricky than seed-based analysis, the interpretation is going to be a little bit more complicated as well. And this is a review article that you can look into if you're interested in reading more about the kinds of issues with both of these approaches, seed-based correlation and ICA. The third class of methods we'll talk about is called complex network analysis. And this is based on methods from graph theory, where the building blocks are nodes and edges. So in fMRI data, we typically use brain areas as nodes, and the edges will reflect some estimate of their functional interactions, like their correlation strength. Once we've specified these nodes and edges, we can try to characterize properties of either the overall network organization or properties of individual nodes. So for example, in terms of their more global organization, we can ask questions about whether groups of nodes tend to be highly interconnected within themselves and perhaps less connected between different groups, similar to communities in a social network. Or you might focus on individual nodes and ask whether certain areas seem to be acting like as hubs or bridging distinct communities, which in terms of brain function has been speculated to relate to integrating or associating between different modalities. There's a lot of different possible measures that people are using with this complex network analysis, and just a couple of references if you want to learn more about that. One of the key parts of this analysis is how we choose our nodes. This will really affect whatever we get from the data. And so if we're defining nodes for a whole brain analysis, we'd like to carve up the brain into kind of meaningful functional units rather than arbitrary boundaries. So if we just randomly carved out boundaries, then at the end of the day, we'd run some analysis and get results that might give you no information at all. And so if we're looking at functional networks, then maybe one thing we might be interested in is to have nodes whose the time series within the node are homogeneous. There have been a lot of different methods working on different ways of parcellating the brain into different functional boundaries. Okay, so the next section will cover some points about the interpretation. And I think that really the main message I wanted to convey here is that resting state and functional connectivity seem to be powerful in that they're helping us to uncover a lot of interesting phenomena that often relate to other things we measure, like behavior and disease. but it's actually really hard to clearly interpret what we're getting because there are so many ambiguities and it's not clear exactly what our signals mean. So in resting state scan, our data are going to reflect a mixture of many unknown processes that are going on, and we don't have a clear understanding of all the factors that are shaping what we end up observing at the end of the day. The relationship to the structural connectivity as well as the electrophysiology is not a clear picture yet, and the data are noisy, and the results can be quite sensitive to the modeling and pre-processing decisions that we make. So with regard to that last point, artifacts like head motion have to be treated very carefully because even small amounts of head motion can impact your resting state measures. So this would be especially an issue if you would expect there to be any systematic differences in head motion, like across age groups, if children might be moving more than adults, which was actually shown here. And this was found to cause spurious age effects of functional connectivity, and it really masked any real neural connectivity effects. Another one is that systemic physiological processes like breathing and cardiac activity will also have large and widespread relationships with fMRI signals because they alter quantities that are very relevant to bold contrast like cerebral blood flow or blood volume. And so, for example, Rasmus Byrne and others have shown that changes in the rate and depth of breathing over time, known as RVT, correlate with bold signal across much of the gray matter. And of relevance for functional connectivity, it's important that since it's modulating many areas of the brain in common, then this could create spurious correlations that we might get from a connectivity analysis. Because if we were to put a seed in an area that has a lot of respiration effects, then it would tend to pull out a whole bunch of other regions that are also showing respiration effects. So we want to try in general to record and minimize these effects so that you can try to understand if that's impacting your results or to try to clean up your data. And I won't go into any more detail right now because there was already a lecture on this a few weeks ago, I think, by Dan. But I did want to say that in resting state, distinguishing signal from noise is even harder than when you're doing studies of task-related activity because we have no model that helps us distinguish it, and we can't do things like average across trials to try to improve our signal-to-noise ratio. And when we're doing functional connectivity analyses, what we're doing is quantifying the relationship between brain regions, and each of these brain regions is a mixture of signal and noise. And that's a bit different from taking our standard task analyses where we're comparing our data to a noise-free activation, the regressor. And so this can create more danger in drawing the wrong conclusions about brain networks, especially if we don't treat artifacts like from physiological noise or head motion. As I mentioned before, our results can also be sensitive to the type of pre-processing that you're going to apply during your scans. And one thing that's tricky is that it's also hard to evaluate whether your pre-processing strategies, whether one given pre-processing strategy is actually doing the right thing, because we don't have an absolute truth in many of these cases. And so perhaps the most famous example of this in the resting state literature was the so-called saga of the global signal, where in 2005 there was an important study showing that the default mode network had negative correlations with attentional networks at rest. And that was interesting because it was thought to reflect some kind of intrinsic balance between externally or internally focused systems. But here they used a pre-processing step known as global signal regression, and that's where you take the average time course of all the voxels in the brain and you regress that out of the data. So it's basically an aggressive way of trying to remove anything that might be common across all of the voxels. But it was pointed out that this could mathematically introduce negative correlations in the data, and other people had some trouble replicating the anti-correlation findings without applying global signal regression. But how do we know whether we should be doing it or not? And there's still a ton of debate about this, because on the one hand, global signal regression may be too aggressive and creating these extra negative correlations. But on the other hand, if we don't do that, it could leave too much common noise in our data and also prevent us from detecting any real negative correlations that might be there. So it's been shown later on that anticorrelations are present without global signal regression if we use methods that like physiological noise reduction to try to mitigate these common influences of heart rate and respiration, or to target tissue types like the CSF, where you might not expect neural activity. And personally, I prefer to try to use methods that target specific types of signals that we're measuring and model those out of the data, rather than to just regress out the whole brain average, since that average signal is kind of ill-defined in the first place. I mean, in any given scan, it could reflect a totally different balance of effects. So in one scan, it may be like dominated by physiological noise. In another scan, you may just be getting more contributions from brain networks that are more highly active during that time. But this is all still really, really controversial, and there are a lot of different viewpoints. So a neural effect that can impact fMRI signals globally is fluctuations in alertness or vigilance levels. So in resting state especially, people can get really sleepy during our scans. And so unless the goal is to study alertness in itself, then this might end up being a confound in your study. And there have been effects in fMRI data shown to happen when people become drowsy or fall asleep. One is that the fMRI fluctuation amplitude tends to increase a lot, and we tend to see stronger global fluctuations across the cortex, which may be due to common neural modulation by subcortical systems that control brain arousal. So if you have a drowsier subject, you may see more global signal. And in order to tell if someone's becoming sleepy during your scan, the best way would be to record something like EEG during the scan, but that's not always feasible, and a lot of the big fMRI data sets don't have that. So one direction that several people have been pursuing are to develop methods to try to infer the vigilance levels of the subject from the fMRI data itself. So that might be helpful for trying to sort out some of these state-related effects. There's also some studies showing that depending on what you're thinking during the scan, this could affect your correlation patterns and signals that you obtain. So here they found robust differences in the pairwise correlations that depended on whether they had a subject imagining a song, I think, during the whole scan versus counting backwards in their head to try to represent kind of typical things people might do in the scan if they're bored in a resting state study. And in some more recent work, Javier showed that we might be able to use unsupervised machine learning techniques to try to detect which of several brain states a person might be in during some window of time during your resting state scan. In terms of the correspondence with the anatomy, it's possible, it's very likely that the anatomy will exert a strong influence on the types of patterns of functional connectivity that we see, since it's constraining the propagation of activity in the brain. And there's a lot of studies that have demonstrated that there is a strong correspondence between functional and structural connectivity. Here they found that regions in the cerebellum tended to show very specific functional connectivity to areas in cortex that they were known to project to, and different subregions of the thalamus were found to preferentially correlate with their areas in the cortex that they target. And we can also probe this correspondence by trying to study clinical populations that lack certain connections. And in this case, patients born without a corpus callosum still showed bilateral task activation, but their interhemispheric correlations in resting state were reduced. And here was a study of patients that had surgery to remove corpus callosum, and they found that after the surgery, there was a disruption in this bilateral correlations, but that some areas still had more preserved bilateral correlations, and they had mentioned there that these areas had connections that didn't cross in the corpus callosum. So there's not a one-to-one relationship between structural and functional connectivity, and you can imagine that if you have two regions that don't have a direct connection between them, but they both have a connection to a third common area, that area might be driving correlations between them. So we can't directly use functional connectivity as a proxy for structural connectivity. To better understand our signals, we also want to understand how they relate to electrophysiological measures of neural activity. And to look at this, we could record neural activity or electrophysiology simultaneously with fMRI, either non-invasively in humans with EEG or more invasively in animals or certain populations, patient populations. One of the findings that has emerged here is that spontaneous fMRI signals correlate with fluctuations in the broadband gamma power of electrophysiological signals. Not only that, but the correlations between gamma power fluctuations measured at different sites in the brain, here with ECOG grids, these seem to align closely with fMRI correlation patterns. So one reason it's important to kind of see these findings is that it's evidence that the fMRI correlations that we're seeing may not be may be related to correlated neural activity and not just emerge from factors like vascular or systemic physiological effects. But the exact mapping between features in the electrical activity and fMRI is not fully resolved, and a number of different features in the electrophysiological signal have been found to correlate with resting state data, so this is still an evolving story. Okay. So as an overall summary here, resting state data exhibit these spatial temporal correlations, this very rich organization that's being studied by many people for clinical applications and basic neuroscience. There are many ways of analyzing the data, a lot more than we talked about here, but it's really important to be aware of how these methods are working and their trade-offs between them, and to try to stay as close to the data as you can. So like look at the raw time series a lot and try to understand like if regions are more or less correlated, is that because their synchrony is higher, because the SNR is better, the amplitude is higher, all these other things that interrelate. And be careful because, you know, terms like network and connectivity will really depend on the study that you're reading. And so there are many different factors that can introduce variability in our signals and connectivity measures. So it's important to try to understand those and control for things as best as possible. The interpretation is still not clear, and more work is needed to understand the neural and physiological basis so we can try to use these signals more effectively. And in the last few minutes, I just wanted to touch on a couple of future directions in these areas. And on the analysis side, there's still a lot of work being done in terms of how we can better analyze and extract information about the spatial temporal dynamics of brain activity, not only in resting state, but also in task data. Just one area is this growing direction of time varying analysis. So instead of just making one summary map of the data over a whole scan, which is typically done, if we take smaller chunks of data and look at how features vary across the scan, could that give us perhaps any additional insight into either brain network function or cognitive states or brain disorders, for example? And so here it was shown using a schizophrenia population that the amount of time that someone spent in a given connectivity state or more transient correlation pattern, this was more sensitive in indicating schizophrenia compared to looking at the average connectivity differences across the whole scan. And this is just one example, but there's a lot of development and applications along different lines. A second one is how are we going to improve our knowledge of neural and physiological basis of resting state signals so we can really use this data much better and understand it. And this could come from trying to do more multimodal recordings, so acquiring complementary information like electrophysiology or EEG during the scan to understand the brain states and the concordance between these different signals. Or by doing experimental manipulations where we try to causally activate or inactivate certain specific brain regions and then look at what happens across the rest of the brain. So this could give us some more understanding of the kind of signal origins. Or to look at the dynamics of the vasculature at high resolution and understand how these hemodynamic fluctuations may contribute to the resting state activity that we're seeing. a lot of work is also trying to push the limits for, push the boundaries for clinical applications and in trying to design more specific and sensitive biomarkers, which could be useful at the level of the individual subject, or perhaps to help us define subtypes of neuropsychiatric disorders such as depression. And I think it would be helpful if we can integrate all of the information that in these kind of research directions into this effort as well. And lastly, data sharing has really transformed resting state fMRI. You can just download a whole bunch of data from the web now and just mine it and find many interesting things that you can't see when you have much smaller sample sizes. And so that's very promising. And that's all I have for now. So thank you so much. Yeah.
So speaking of sample sizes, can you give us some rough guidelines as to like the size of the population that you might need, depending on how many minutes of resting state you left, what kind of analysis you want to do, ICA versus investigation? Are there any guidelines for that?
So the question was, there any I have to repeat it because it's being recorded. Are there any guidelines as to the sample sizes you should use when you're trying to discover various effects using these seed-based or ICA analyses? I would say that, I mean, it's sort of similar to task-based studies where, I mean, we almost never have enough subjects, I could say, or enough scan time. In terms of, so I don't really have a really good answer for that into things that are specific for resting state fMRI, and it will, of course, depend on the effect size of what you're trying to find. But scanning longer and longer is actually becoming one direction people are pursuing because you can kind of sample more brain states, and that might give us useful information about brain dynamics. So if you just capture 2 to 3 minutes, or well, no one does that short, but like 5 to 6 minutes of someone's brain activity, then you're just capturing them in one particular state, and there may be a lot of individual intra-individual variability depending on when you happen to capture that state. So yeah, so I guess longer scans and more subjects are better, but the number of subjects I tend to see in resting state studies are kind of along the order of task studies, except with these new databases like the Human Connectome Project and the Biobank, which, I mean, resting state, it's so much, I guess, easier to share data in the sense that everyone's doing the same thing in the task, supposedly. Actually, that's an oversimplification, but if you can definitely download, like, you know, a lot of subjects and try to find effects that you may not be able to see in smaller numbers of subjects. Yeah.
So the pre-processing pipeline seems a little dark magic, and I was wondering if there's any kind of attempt to standardize this pipeline.
Yeah, the question was, is there any attempt to try to standardize pre-processing pipelines? Yeah, there actually there are some efforts out there. I blanking on the name right now, but there are people who have been trying to really standardize the data format. I think from Russ Poldrak's group and others, and try to produce ways of being able to more to better replicate your results and other people's results. But the order of the pre-processing steps is, I mean, it still faces the same issues as fMRI. Some people do things in a slightly different order. There are certain steps that shouldn't be done before others, but other steps where it's not necessarily clear what is better to do. So there's still a lot of work on comparing different pipelines. Okay.
I was wondering if you could comment on trying to pull data acquired from different sites. So there's a lot of public data sets out there, and unlike structural or diffusion imaging, where you have a different scanner, it's hard to compare the measurements because you actually get systematic differences. And at resting state, it's kind of self-normalizing because you're actually doing correlations against yourself. So perhaps you can compare things across different scanners. But I was wondering if you could comment on.
Yeah. The question is like, how can we just readily compare or pool data from different sites in resting state? Is that easier than some other? So I think so. I haven't really personally done this, so I can't really. I don't have a great sense of how reliable it is across different sites. If you're comparing correlations, as you said, do things normalize? I might have to think about that one a little more. Yeah, I mean, even though people are not doing anything, there can still, in terms of there being no task, there can also still be variability in the pulse sequences and the resolution and the scanners that can show quite a bit of difference. And I don't know how much difference that plays into resting state measures, but that's a good question.
It seems like in the literature, you see that more often where people pull
Yeah.
data other than other types of.
Yeah, yeah, and I think it's probably it may be because, well, there's a lot of different task databases as well, but I guess trying to because tasks probably vary a lot more across sites than, and depending on what the experimenter is trying to test compared to resting state, we think, oh, people are just lying there with their eyes closed, in the scanner, so we can try to combine different sites. So that's probably why we see it more, but it's a good question as to how much that site effect matters, or acquisition.
Actually, when you say that it agrees well with that beta band coherence, the resting state, are you talking about the whole network or just particular link that agrees? It shows correlation here and there, but can you reconstruct the whole network?
So that finding with a gamma band power, maybe.
I thought you said beta.
Oh, if I did, I apologize, because I meant broadband gamma power. So this broadband gamma power has been shown to relate to the fluctuations that you get in fMRI, and also to the kind of correlations. In terms of other bands, it's actually, there's quite a lot of inconsistency across the literature. Bands like alpha and beta have been shown to correlate with the default mode network, but it's sort of not so specific that you could actually reconstruct a network from the electrophysiology, because they may correlate with other networks as well. And I think it'll depend a lot on brain state. So if somebody's drowsier, then what you get with alpha power or beta power may be slightly different than if somebody is more alert.
Also, what kind of data was it? Was it MEG or EEG or some direct reporting?
So in one of the figures I showed, that was ECOG data. Yeah, but there have been both EEG and MEG studies trying to look at the correlates of various resting state networks, and it's still kind of a mystery. That's right. Yeah, these were kind of had a lot of coverage, so they were able to look at much of the brain, but not, it's limited compared to fMRI. Yeah, insofar as they could test that, that's true. That's not that slide I show is just kind of a general idea that, yeah, because it's not fully sampled, so we can only test whatever we have there. Yeah. Okay, thanks, guys.